Sebastian Nobel from Mileti and Milteni Biotech here in Germany. And we are going to start with the first speaker, who is Tal Devir from Tel Aviv University in Israel, that is going to speak on personalized tissue implants from 3D printing to bionic tissues. Please.
Thank you very much. Thanks for the introduction and for the organizers for inviting me. Can you please switch to my presentation from my computer? Okay, thank you. So my lab at Tel Aviv University is working on tissue engineering, and we're working on engineering different types of tissues. We work on the heart, on the spinal cord, on the brain, intestine, the retina, and I will give you today an overview of the things that we're doing at the lab. So what is tissue engineering? What do we do? So in this approach, we can take cells from the patient, preferably from the patient, because we don't want to induce immune response. And we cultivate the cells initially in 2D flasks to expand them. Then we need to create tissues from these cells. And tissues in the body are not just the cells. The cells are very important, but the extracellular matrix is important as well. And it serves as a glue to the cells, allowing them to have the function. So we're working on different types of biomaterials. We can add growth factors, small molecules, nanoparticles, and essentially we're doing everything we can to allow the cells to assemble into functioning tissues. And once the tissues are functional, we can take them back and transplant to the patient. So how do we create the tissues in the lab? So we can use different types of cells. So we can use cells from biopsies or adult stem cells. But if we're talking about neuronal tissues or cardiac tissues, we cannot use these cells because they do not differentiate to these type of cells, or we cannot spare these cells from the patient's organ. And in the lab, we're working with induced pluripotent stem cells. We manipulate the cells, we seed them in the biomaterials, and long ago we've shown that we can use such approach to create cardiac patches that were used to regenerate the infarcted heart in animals. But there are still many, many challenges, and one of the challenges is how to create a biomaterial that really mimic the extracellular matrix of the tissue. And for that, what we've initially done, we took pig's hearts, we removed all the cells, and we've left only with the biomaterial. We preserved the extracellular matrix. And what we were able to see that there's one big mass of fibers. There were microfibers, nanofibers, aligned fibers, randomly oriented fibers. And initially, we wanted to evaluate in the lab what's the role of the different fiber populations. And for that, using electrospinning, we've created the microfibers, nanofibers, aligned or randomly oriented fibers. And we've seeded cardiac cells on these matrices and evaluate their role. So this is just one example. You can see that on the aligned fibers, the cells are elongated and aligned. And this is extremely important when we're talking about cardiac tissues because we want to have an anisotropic tissue that the electrical signal will be conducted nicely. And here on the randomly oriented fibers, we do not get such morphology. Another thing that we've investigated was what the effect of the fiber diameter. So we created the nanofibers, microfibers. These are 1 micrometer fibers and 5 micrometer fibers. And usually these days, if you write the word nano in a grant application or in a paper, you increase the chances of acceptance at least in 50%, right? 10 years ago, it was a 100% increase. But actually, the cells do not like the nano-sized fibers. I'm talking about cardiac cells. They're still rounded, and they prefer the bigger fibers. So if you take the morphology into account and you consider also the biochemical content of the matrix, the mechanical properties, you can create a cardiac patch. This is a few centimeters in diameter, few millimeters in thickness that contracts nicely in the lab for a long period of time without external stimulation. However, when we're talking about humans patches that can regenerate a human heart, which the wall thickness is about 1 centimeter, we cannot engineer such patches, because if we're using thicker biomaterials, there's a mass transfer limitation. Oxygen cannot penetrate into these tissues, and the core cells are dying. So what we've developed is a way to create many layers of tissues, for example, grooved layer for cardiac cells, and channeled layers with for endothelial cells to form blood vessels, and honeycomb structures to release VEGF from these particles, and even honeycomb structures to release dexamethasone, an anti-inflammatory drug. So we can grow these tissue layers in parallel in the lab, provide the best conditions for each layer, and then just prior to transplantation, assemble these layers and transplant them. And we can see, this is just one example, that once we release VEGF, you can see nice vascularization penetrating, blood vessels penetrating into the patch. But when we're talking about biomaterials for tissue engineering, initially people thought about synthetic materials, and synthetic materials, once you introduce them to the body, they provoke some kind of immune response. And if we're talking about the heart or the brain, we don't want to have a fibrotic tissue around the tissues that we're transplanting. So people thought, well, let's take pigs material, remove all the cells, and we're left only with the collagen fibers of the pigs, and these will not provoke an immune response. So what we've recently shown that there are certain antigens in pigs that we do not have. For example, this sugar alpha-gal that is present in pigs, not in human, and this will provoke immune response. Additionally, there are differences in the amino acid sequence of collagens and other ECM proteins that we do share with pigs, and these differences may provoke an immune response. So what we decided to do was to rely on the patient's own cells and the patient's own materials. So what do we do here? We can take a fatty tissue from the patient, isolate the cells, reprogram them to become induced pluripotent stem cells, and the exosomal matrix of this fatty tissue is processed to become a personalized scaffold. Then we can put the cells back, differentiate them to whatever cell type that we wish, and we have an autologous patch. And if we look at the immune response to these matrices, what we've initially done was to simulate immune response in humans. I will not go into the details of the experiments. I'll just show the results that indicate that there's a clear difference between pigs materials and human materials, and also that there's a difference in between the donors, implying that there's a need for a personalized material. We've done it also in animals, showing that there's less immune response to autologous materials. So when we transplant, for example, the cardiac patches, you can see two things here. First, that there is an open chest surgery, and the second thing is that we have 2 stitches here to secure the patches to the heart, and I will address the 2 stitches in a few slides. But the open chest surgery is not an ideal thing for a patient who just had the heart attack, and what we've decided to do is to take the patient's own materials and create a material that is liquid in room temperature. Now we can mix the cells here, and in 37 degrees it solidifies. So now we can take this personalized hydrogel, inject it wherever we want to the body, and it will solidify and will create a tissue in the body. So what can we do with this technology? So we can isolate the cells, take the stromal cells, reprogram them to become induced pluripotent stem cells. The ECM is processed to become a personalized hydrogel. And once we mix the cells with the hydrogel, we can differentiate the cells inside a 3D network of fibers to any tissue type that we wish. This is just a partial list of the things that we're doing in the lab. For example, adipogenic implants for reconstructive surgeries, motor neuron implants for spinal cord injuries, cortical neuron implants for brain trauma, or dopaminergic implants for Parkinson, and cardiovascular implants for cardiovascular diseases. And we can also use more advanced technologies such as 3D printers and microfluidics to create more complex tissues. So just to show you that these cells were used to be fat cells a couple of weeks before, and now after 14 days they became cardiomyocytes, functional cells. The implants are contracting, firing, as you can see here. And when we inject these implants to animals' heart, we can see that we can regenerate the infarcted heart of small animals. We've done the same thing with the big animals, with the pigs, injecting them to the scar tissue. And these are preliminary results showing that we can regenerate or have a beneficial effect of the treatment. So we're also using the hydrogel, the personalized hydrogel with the patient-specific cells to 3D print patient-specific tissues. So we look at CT images of the patient, we look at the blood vessels, we transfer the blood vessels to the computer, we add more vessels to better nourish the tissue that we're printing. Now we go to the printer, printing the myocardium and the blood vessel forming cells together, and everything is in patient-specific materials and with patient-specific cells. Eventually, it looks like that. So it fits not just the cellular properties and the biochemical properties of the patient, but also the anatomy of the patient. And this is just a cross-section of one blood vessel within these printed patches. You can also see the transfer of the electrical signal. This is one blood vessel, this is the entrance of the vessel, and the tissue contracts nicely. So we have a method to create, to 3D print patches, and we've already started working on printing whole hearts with blood vessels. This is a very basic heart, but it is from the patient's own materials and the patient's own cells. The cells contract within these hearts, but we now need to teach them how to contract together to have a pumping ability. And this is in the middle of the printing. You can see the different chambers and the blood vessels, and this is at the end point. So we can do the same thing for spinal cord injury. So instead of differentiating the cells to cardiomyocytes, we're differentiating them to motor neurons. We create incisions in animals, and we transplant these tissues. And I'll jump to the movies. This is first in acute model. You can see that already after a week, this is the untreated animal. You can see that one of its legs is not really operating. It's not really putting pressure on this catwalk. However, when we're working with the full implant, 100% of the animals, more than 20 animals in each group we have here, 100% of these animals are working perfectly. This is an acute model. And when we're talking about a chronic model, which is more relevant for patients, we can see much better effect of the full implant compared to the other groups. Sometimes we don't need a full patch or a full implant for the patient. For example, in Parkinson, we just need to inject cells to the substantia nigra. However, when we do that, most of the cells are dying because they do not form cell and cell matrix interactions. So what we've decided to do to take isolated cells inside the hydrogel and create droplets of cells. So we're injecting the hydrogel with the cells here. Here we're injecting oil. So we can create droplets. We heat it up to 37 degrees, and we can collect these droplets, and now we can inject them wherever we want in the body, and we're doing that in Parkinson models in animals right now. So we can create cortical implants with the same procedure, and dopaminergic implants, as you can see here. These are 90-day implants in the lab that they secrete dopamine, and as I mentioned, we're now transplanting these patches in animals. We also create mesenchymal stem cell encapsulated in hydrogels to treat age-related macular degeneration. So we create we encapsulate the cells, and we're injecting them directly to the retina. This is now in process. Now we're back to the heart. Another issue that we have with the heart patches is the transfer of the electrical signal. We're culturing the cells inside biomaterials that are not really conducting. And what we've decided to do is to incorporate gold nanowires into the biomaterials to help the cells interact with each other on the electrical level. And here you will see 2 movies. This is without gold nanowires. The cells are not contracting synchronously, but once we have biomaterials with the gold nanowires, you can see that everything is contracting together. And this is already patient-specific materials without gold, 4 nanometer gold particles, or 10 nanometers. And you can see here that we've taken biological material, and now it's fully conducting with all the biological motifs. And it works also with neurons. So this is after 3 days in culture. You can see the extensions of the axons with the gold nanoparticles compared to the pristine scaffolds. Also worked with the dopaminergic implants. They secrete more dopamine when they're cultured in materials with gold nanoparticles, meaning that they can transfer the electrical signal better and then secrete more dopamine. So if you remember the 2 stitches on the heart, which is not really ideal for the patient, we decided to exploit other properties of gold nanoparticles, or gold nanorods in this case, to fix the patch onto the heart without stitches. So gold nanorods, they have nice optical properties that if you illuminate them in near IR, they convert the light to thermal energy, and then you can really solder the tissue to the heart. And this is what we've done, 30 seconds of illumination, and now the cardiac patch is stuck on the heart, is not cannot be pulled off. This is by electron microscopy. You can see the heart, you can see the patch, and the integration point. Last family of projects, what we call cyborg tissues, the integration of electronics, nano and microelectronics with engineered tissues. Not just because it's fun and nice and cool to integrate electronics with living systems, but because there was a need to sense the function of the tissues online, and we've created these electronic spores, electronic systems, incorporated them with the 3D scaffolds to have 3D scaffolds with many, many nanosensors within. Once we culture cardiac cells on these matrices, and this is just one electrode, but we have many, many of these electrodes, we can sense the function of the tissue. Online, we can add drugs such as noradrenaline and immediately see how the tissue reacts to that. So it was a nice publication and a good patent, but we weren't really happy with that because in this case, we couldn't do anything if the tissue was not working properly. We couldn't interfere. So we designed the next generation of the cyborg tissues that in addition to the sensing, we can provide electrical stimulation or release drugs. So we can just type on the computer and provide stimulation to pace up the tissue or release different drugs according to the sensing that we're doing. So if we sense that there is not enough oxygen, we can release, for example, VEGF or SDF1 that can induce new blood vessel formation, or if we sense that there is inflammation, release anti-inflammatory drugs, and these are things that I'm not imagining we've already done. So we can also create flexible and stretchable electronics. We can release multiple drugs in parallel, and we can also reload drugs into these electronics just by injecting it to the circulation. However, when we're talking about electronics in the body, which we cannot use electronics that are made by lithography from the microelectronic industry because they do not have the same mechanical properties and our body is dynamic. So there is a mismatch in mechanical properties and this will induce immune response. So what we've decided to do was to 3D print the electronics with the matching mechanical properties to the tissue, and this is in the patient-specific hydrogel and with the patient-specific cell. So we print these electronic systems, and as you can see here, the electronics are very soft. We can manipulate them. They have the same mechanical properties as the tissue, but we can also use them for recording of the function and for stimulation. We sometimes don't want the electronics to stay there for a long period of time, so we created degradable electronics that in certain time will just eliminate from the body. And my last slide, because I see that people are very punctuate here. So we've created the whole hearts now with the electronic on strategic areas on the heart. For example, we have stimulations on the septum. We have a release of drugs on the left ventricle and other things that we're doing. And just to acknowledge my group members, collaborators, and funding. And if you have questions, I'll be happy to answer.
Thank you very much for the very nice talk and for staying in time. We have time for one or two short questions from the floor. One there, please.
Thank you for the presentation. Concerning the application for the nervous system, if you understood well, you are using omentum as a source of hydrogel. I wonder if you are thinking for different sources, since the extracellular matrix composition of the nervous system is completely different from peripheral tissues, including omentum.
Yeah, it's a very good point. What we are able to see first that there is the omentum is a tissue with remarkable regenerative capabilities. And the second thing that we were able to see that once we culture the cells and differentiate them, we mimic the developmental stages of these cells in vitro, and they completely replace the extracellular matrix with a matrix that is suitable for them. For example, for cardiac cells, we see ECM that is associated with cardiac cells. With the neuronal tissues, we see ECM proteins that are only present in neuronal tissues. So, as a starting point, this is a good material to start because it has the regenerative capabilities and it serves as a depot for stem cells in the body. It also serves, it can also be taken from patient with a minimally invasive technique. So it's a perfect material, starting material for us.
Any other questions? Can you elaborate a little bit how the material jellifies? What is the mechanism for the
for gelation?
Yes.
So we take the material after decellularization. We cut it to small pieces, and then we have pieces of collagen fibers. And once you heat it up to 37 degrees, the fibers are entangled together, and it's a physical cross-linking, which you can apply energy to break it. You need to cool it down and then apply energy to break it, but it will not just break down like that.
And mechanically is enough, the physical cross-linking?
For soft tissues, it is. And again, while you differentiate the cells inside this hydrogel, they enforce the hydrogel because they secrete more ECM proteins, and then you eventually have a tissue with the perfectly matching mechanical properties.
Okay. Thank you very much once again for a very nice talk.
Thank you.
Okay, I'm happy to announce our next speaker. It's Eric Schifut. He's postdoctoral researcher at the University of California, San Francisco, and his talk is entitled Genome-Wide CRISPR Screens in Primary Human T Cells Reveal Key Regulators of Immune Function. Welcome.
Hi, everyone. It's a real pleasure to be here. I would like to thank the organizers for the invitation. Although I'm representing UCSF today, I was trained at the Weizmann Institute in Israel, and thus it's also a unique pleasure to both follow and precede members of my home country. So my goal today is to introduce you to discovery platforms that we developed to study primary human T cells. My work aims to understand how human T cell function can be engineered to become more productive cell therapies. It is clear that the cancer immunotherapy has revolutionized the oncology practice. In particular, engineered cell therapies represent a fundamentally different way of treating patients. In such therapies, cells are extracted from patients' blood, and then they're engineered, usually using an antivirus, to introduce a new gene, a new feature, or a new targeting ability of these cells. Then these cells are massively expanded ex vivo and infused back to the patient. Many components of this system were also engineered and designed, developed in the Weizmann Institute in Israel, and it's, again, a point of pride. This technology now is transforming the lives of many patients, as I mentioned, and here is one cohort to illustrate that point. So here I'm showing a cohort of 56 patients diagnosed with CLL that was otherwise refractory to other modes of therapy. On the vertical axis, I'm showing the patients that responded well to this therapy throughout the follow-up of this study on the horizontal axis. The first statistic to be extracted from such a survival curve is actually quite grim. In this particular case, only 20% of the patients responded well to their engineered cells. This gap in therapy represents a gap in our knowledge of how to design more effective cell therapies. However, the second statistic to be extracted here is actually very promising. For the subset of patients that did respond, their response was long-lasting and durable. In 20% of these patients, their engineered cells have protected them from relapse for more than 5 years. The follow-up is still ongoing. This represents a new class of drugs. These are programmable, self-amplifying, living drugs that persist in the patient's blood. This new class of drugs demands a new strategy for drug discovery, and this is exactly my mission. Today, I will introduce you to an experimental and computational platform that allows us to accelerate the design of next-generation cell therapies and hopefully overcome the existing challenges. In order to design more effective cell therapies, we must first understand what makes an effective cell therapy. So here, from the same cohort of CLL patients, I'm showing stratification of the patients in two groups. Those that responded to the therapy on the left, and those that did not respond on the right. On the y-axis, this is a measurement of the expansion of their engineered cells. So we can clearly see that one critical feature of the success of these therapies is the initial proliferation of the infused cells. Moreover, in a very interesting case, in one particular patient in this cohort, the researchers found a very interesting phenomena. So these pie charts represent the repertoire of these engineered cells. Each color represents a different variable segment. Before infusion, the patient's repertoires are quite diverse and represent almost all variable segments. But throughout the study, the researchers found that the engineered cells were dominated eventually by a single dominant V segment. When they asked why this is happening, they sequenced the genome of this particular clone once isolated. Then they found that through happenstance, the CAR construct was integrated into a TET2 gene. This TET2 gene disruption, together with a hypomorphic allele in this particular patient, created essentially TET2 knockout cells. These cells now dominated the response and led the response in this patient. This very bizarre and peculiar experiment shows us two things. One, with a specific knockout, we can engineer cells that eventually will have an increased proliferative capacity. And the second thing is quite tantalizing. In this particular patient, it might be that one engineered cell has led to this remission. So now, knowing that proliferation is key for the success of these type of strategies, I wanted to ask the question, what genetic programs regulate T cell proliferation? And also, we wanted to approach this in the most unbiased way possible. So the way to go about it is to do some kind of genetic screen for different perturbations. So just as a quick intro, we can divide genetic screens to two types, arrayed screens and pooled screens. In arrayed screens, they're quite simple to understand. Basically, each well will have a homogeneous population of cells, all knocked out for the same gene. You can see the description of that here on the right. For pooled screens, we actually introduce the perturbation using lentivirus. This relies on two technologies. One, our ability to synthesize oligos in large scales, short oligos. Then cells are all cultured in the same vessel, applied some selective force, and then we sort on some marker of choice. Using next generation sequencing, we can sequence the guide barcode, and that tells us which perturbation each cell has undergone. These pooled screens have been remarkably powerful in discovering fundamental genetics and mouse immunology. However, so far there have been a requirement for these pooled screens that the Cas9 will be stably expressed in T cells. So that has not been possible or very challenging to do in primary human T cells because the Cas9 virus is quite large and the transduction efficiency is very low. However, when we do Cas9 RNP or ribonucleic protein electroporation, where we just couple a synthetic guide RNA and a synthetic protein, although we can perform a successful knockout, this is only compatible with the arrayed screens because the guide will eventually be degraded and we'll have no record of what the perturbation the cell has undergone. So to solve that problem, we actually took a hybrid approach and created the best of both worlds. In such a platform, cells are extracted from patients' blood. Then in this particular cohort, we'll study CD8 T cells. We introduce the guide virus, which is small, compact, and introduces quite efficiently using lentivirus that will integrate into the genome of the T cells. And the Cas9 will be electroporated. So our question basically was, can we find the Cas9 complex together with the transduced guide and eventually lead to a functional knockout? So here I'm showing the result from one of our first early attempts. On the y-axis, you see a marker that we chose to edit, the CD8 receptor itself. And on the x-axis, this is a fluorescent reporter for transduced cells. So right after Cas9 electroporation, we can see that the editing of this marker has been reduced to almost none in all cells that produces the guide. And it's also specific, so cells that don't have the virus remain at wild-type levels. This is not specific for this particular receptor and not only for CD8 T cells. We show this for many markers. Here is another one, CD45, showing across multiple donors that we're able to edit the protein level. So what's nice about pool screens is once it works for one guide, it actually works for a pool of guides. So all we need to do to go from this pilot experiment of one guide is just produce a library that has guides targeting the targets of choice. Through a series of optimization, we actually scaled up the system that we term slides for guide RNA lentiviral infection with Cas9 electroporation to a genome-wide library. This genome-wide library is available on Agene as a resource for the scientific community. It's composed of 80,000 guides, more or less, targeting 20,000 protein-coding genes. We did this screen in 2 individual donors. We introduced the library as I've shown you before, electroporated the Cas9, expanded the T cells, and then re-exposed them to TCR stimulation. In particular, as I mentioned, our goal was to try to find regulators of T cell proliferation in response to stimulation. So thus we used a CFSC, which is a cell proliferation dye that dilutes as cells divide. Based on that CFSC proliferation curve, we sorted the cells into 2 bins and then asked which guides are enriched in the dividing cells and which are depleted. So When I show you the log-fold change values, positive values will mean that this guide, this knockout, is enriched in the dividing cells. So here I'm showing 3 exemplar positive hits and 3 negative hits. CBL-B, CD5, and UPASH3A are all known negative regulators of T-cell proliferation and T-cell signaling. So here, each line is a guide, and because we have multiple guides targeting each gene, we're confident that these are true hits because there's multiple guides that are targeting them that are enriched in the positive population. On the other hand, to disrupt TCR signaling, to prevent cells from responding to the signal, we can also do that using knockout of essential T-cell signaling genes. As a reminder, we're not doing this screen only on these 6 genes. We're actually doing it on all protein coding genes. So now we can create these unbiased maps of T-cell signaling. So this is one of the first times that this has been done in primary human T cells. As I mentioned, it has been challenging so far because there was no way to introduce the Cas9 using lentivirus. So now we get these 2 arms of the screen, those that are, if we knock them out, will make T-cell proliferate more strongly, and those that, if we knock them out, will prevent T cells from responding. So although there's many hallmark genes in there, and it's not very particular to go to any one group, we would also wanted to see if whether the screen is just an artifact of these 2 donors or is it repeated. So here I'm showing the correlation between the negative and positive hits of 2 replicates. Each one has been done in 2 individual donors, and we can definitely see that the targets tend to repeat themselves. How do we annotate those hits now once we have them? The negative hits are very easy to annotate because T-cell signaling has been studied for many years. So using gene set enrichment analysis, we can easily see that our hits are hitting the right pathways. However, for the positive hits, things that if you knock them out, T cells proliferate faster, those are harder to characterize. So to do that, we went to the literature and found a pooled screen, and it was done with shRNA technology that basically injected tumor-specific T cells that were introduced with the shRNA library and asked which genes are enriched in the tumor versus the spleen. So quite strikingly, the hits that we got from the in vitro human model were very enriched in the mouse in vivo study. As I mentioned, this is one of the only enrichment that is possible because these genes, the negative regulators, are not necessarily all concentrated on the same pathway. They come from a diverse set of annotation. Okay, so we have hits. Now we want to first see that we can validate them. So we can take the same technology, but now instead of introducing the guide with lentivirus, we'll just use synthetic guides that are easy to get. And we'll go to the phase of the platform that is the array screen. You got a list of hits, and now we want to validate them. So using arrays of these guides, we first want to see using CFSE, which was the same marker that we used for the pool screen, that guides indeed increase the proliferation of T cells. So for 2 hallmark genes here, across 2 guides targeting them, across 2 donors, we can definitely see that the proliferation is increased once they're knocked out compared to a non-targeting control guide not present in the human genome. On the other hand, our negative control, or an essential member of the TCR signaling LCP2, when you knock it out, cells do not respond to the stimulus at all. So as I mentioned, we do this as an array, so we have a full list of our hits. And indeed, the majority of them recapitulate their ability to increase T-cell proliferation once knocked out. There are some that don't, and there are interesting cases by themselves. So now that we validated the proliferation capacity, we can start to ask what is the mechanism driving this. So first, we can look at classical activation markers for T-cell proliferation, for T-cell activation, CD154, which is also known as CD40 ligand, and CD69. For the majority of hits in our screen, we can definitely see that cells are also hyperactivated compared to the non-targeting control that is here in black. But this is using flow, and it's just for 2 markers, but there could also be other underlying mechanisms. So how can we do this? How can we follow up on the mechanism of each of these hits in a mechanistic way? To do this, we recruit single-cell RNA sequencing and use a technology or approach called perturb-seq or crop-seq that essentially does a pooled CRISPR screen, but instead of sorting on a single marker, now we submit the cells to single-cell RNA sequencing. The trick about this is that the guide will also be captured. So now for every cell, not only that we know which genes are enriched, but also we know which guide we expressed in that cell. So here I'm showing hallmark markers of T-cell state. IL-7 receptor is upregulated, and that will mark the quiescent cells. Cells are not responding. CCR7 in a similar way. Cas67 is a cell proliferation marker, and that will mark the cells that are hyperproliferative, and granzyme B for cytotoxic activity. So as I mentioned, not only we have the gene expression, we also know which guides are expressed in each cell now. So we get this very rich matrix of cells and perturbations, cell state and perturbations. For CBL-B and CD5, we can see that they're definitely enriched in the regions that are also where proliferating cells are. SCP2, if we knock it out, cells do not respond, and they remain in this quiescent population. We can go in a higher resolution and now unbiasedly cluster the cells based on their gene expression similarity. And we'll see that across many clusters, we find typical patterns of guide enrichment in each. This can also now lead you to think that you can start thinking of which perturbations will work synergistically with each other, possibly if they're not using the same pathway. As a thought experiment, this could also be used to start thinking of combinatorial therapies with checkpoint inhibitors and a way to test them all in one experiment. So lastly, the last piece of data that I want to show you is how these cells perform functionally. So you do the mechanism. I showed you that we can go and start coupling the cell state with the perturbation. But do these cells kill tumor more efficiently? So to do this, we need to give them specificity to target the tumor. And we do this using lentivirus. The lentivirus will introduce a TCR that is targeting a melanoma antigen that is expressed on these 8375 cell line. Together with the radar and P editing that we used for the validation, now we have an array of cultural T cells. They are antigen specific and perturbed. And we can layer them on 8375 cells and monitor them through live cell imaging. So first, we see that the killing is specific. As we add more T cells, we see more apoptotic activity using Cas37, and also the cell count of the target cells is reduced. As I mentioned, we do this for an array now, so we have all our targets from the screen, we validated them, and now we want to know if they are acting functionally in this in vitro killing assay. As I mentioned, it's not trivial to assume that proliferation will also lead to increased functional killing. But luckily, we found 4 perturbation, RASA2, CBL-B, SOX1, and TCB2, which actually act in a complex, all to be promoting T-cell killing in this particular assay. Tumor cell killing. And my last data to show you is that just visually, if we just culture the melanoma cell line by itself, it grows uncontrollably. If we add non-targeting control cells, but they're still targeting the tumor, they will clear some of it in this assay. However, our 4 strongest hits, when they're introduced together with antigen specificity, lead to a marked tumor clearance. So to summarize what I've shown you here today, we've used genome-wide screens to systematically map critical genetic perturbations that lead to a boosted T-cell proliferation. With arrays and also multiplex phenotyping using the activation markers, we can see that most of the knockouts do validate, and also they lead to early T-cell activation. The detection of guide transcript using single-cell RNA sequencing has led us to dive down into the genetic programs driving this response. And finally, functionally, I showed you that CBLB, SOX1, RASA2, and TCB2 have all led to increased tumor killing in this assay. So I'll just end by thanking my mentor, Alex Marson, the people involved in this work, our funding resources, and thank you for your attention.
Thank you, Eric, for the nice talk. Are there any questions from the audience?
Hi, great talk. Out of curiosity, did TET2 appear in your screens?
It did not. So our screens are designed to, I can't see you, sorry. The screens are designed to find genes that are contributing to T-cell proliferation only after TCR stimulus. So cells are cultured and expanded after they've been knocked out for about 10 days in culture, even though we don't introduce any TCR stimulus. And then at around day 14 or so, we introduce that TCR stimulation. So TET2 in this case would be a target that would be enriched even without TCR stimulus in our hands. So yeah.
Sorry. Sorry, thank you very much. I have a question. Of course, requirements for proliferation, killing, and so on of different T-cell subsets is different. What kind of T-cells did you use?
So these are CD8 T-cells.
Okay. I mean more central memory effectors or whatever.
We take them as is from patient's blood using magnetically enriched kits. So these involve both naive and memory T-cells. To introduce the perturbation, the guide, and also the Cas9 editing, we expose cells to CD3, CD28 stimulation. So in essence, when we assay the cells, they're all been antigen experienced. And in regards to subsetting the hits for the population that they're relevant to, we think that the single-cell RNA sequencing could lead us to get some hits there.
Okay.
Although this one patient was rescued by probably one T-cell clone here in the back.
Yeah.
It's also kind of worry, right? That in the end, you might end up with uncontrolled proliferation of those cells and even some pre-lymphoma status. So how would you make sure, apart from safety switches, that this is not happening?
Yeah, that's an excellent question, and it's always this problem of the constant struggle between a hyperproliferative tumor cell and what we try to get with these engineered cells. As you alluded to, the possibilities that you have with engineered cells in terms of safety switches are quite flexible. So that's one result or one approach. The other is that for a lot of these, it's a multiple hit models. And with CRISPR, we're very precise about what we're editing. And also, as I mentioned, we try to make sure that our hits are only active when cells see antigen. So those together, hopefully, will narrow us down to a very safe and effective therapy.
Did you make similar screens with regulatory T cells?
With the question was with regulatory T cells, or
no? If you did similar screens with regulatory T cells.
Yeah, that's another possibility. So what's nice about the, again, engineered therapy is that you can move on from thinking of this single type engineered cell and actually engineer an immune system, right? So you can create multiple cell types, each with their function. So there's definitely an option to think of controlling both the gas and the brake here and creating a Treg that will be effective in stopping the response.
Okay, thank you once again. We move to the next talk. Thank you. So the next talk was supposed to be given by Lior Haviv, but he could not make it. So we are going to have instead Zami Eberman, which is in fact the CEO of Pluristem in Israel, and is going to speak on cell therapy makers, opportunities and challenges in manufacturing of cell therapy products. Zami, please. Not this one, huh?
No.
No, we need to get the other presentation. This is not the right presentation.
It should be, we loaded it in the morning, look into Lior Aviv or Zami Aberman.
Do we have the right presentation?
Pardon? We loaded it at 8:30 on your computer.
Okay, here we go.
All right, so thank you. And I would like to take the opportunity and to share with you our finding in the last 14 years, that even though cell therapy is very promising, one of the hurdles is manufacturing. And I think that it's important for us to share the experiment, because I think that one of the promise of cell therapy, especially the allogeneic one, is completely dependent in the ability to manufacture the cells and to enable cell therapy to become an affordable treatment to many patients. So we like to use the slogan here that innovation is a process of turning ideas into manufacturable and marketable form. It's very important because once we think about it, all the observation, how we look on cell therapy in general and in allogeneic cell therapy more specifically, is dependent on a few aspects that are important to mention. First of all, you have to have a good source of potent cells. Second, you have to have trillions of high quality cells, because remember, what we are talking about is allogeneic approach. So we want to guarantee that the same cells will be treated again and again without issues of manufacturing. Then, of course, cost of goods. As you know, it's very doable if one wants to do a product that costs a few hundreds of thousands of dollars to the patient. But if you want to sell a product and make it in a reasonable economical way, you have to reduce the cost of goods in a level that it can be produced. And of course, you have to make sure that you can repeat the manufacturing processes again and again to ensure the quality and what is needed. So you can see here a list of what are the contributes that are required in order to make a consistent product. And one way can say it's not an issue. Each one of the subjects here is easy to handle. We know that there are many technologies existing today. So is it really the case? And the evidence is the newspapers and the announcement. You can see here a list of products that have been approved by the agencies, very famous names, but all of them are facing manufacturing problems. So you have a product, you have an approved product, and then you arrive to the marketing stage that you have to have many products that you can supply, and you face some difficulties. And you see the name, so I'm not going to repeat it. So the goal of a process development step is to transform ideas into product that could be manufactured reproducibly in an industrial process and meet the product and process predominant specification. I don't like to read, but I think that when we did that statement, it was important to us to make an emphasis on each word in that statement, because that's what led our development in the last year. And what we started from the early beginning, I'm talking about 2007, is understanding that once you understand what is your goal, then you can develop a process that will solve the issues that are really important. So we started from the end, and the end is allogeneic cell therapy product that can be manufactured properly And then it brought us to the beginning. And then we start off back and forth development stages, and you can see them on the list. It ends up with a few stages in manufacturing to ensure that we understand what can be produced. We inject those cells into patient, looking back into the data, what have been achieved, understanding what are the issues that we should improve, making a second generation and a third generation, and we are calling them in our facilities manufacturing process 1, 2, 3, and 4, depending on the stage of the company. It represents the 14 years of development, and today we are in phase 3, 2 phase 3 studies that are enrolling hundreds of patients in US, Europe, and Israel, of course. So the source, why we made a decision to look for placenta as a source and not to go to the conventional way, whether to go to bone marrow cells or a umbilical cord blood or other cell source. The main rationale behind that is that the placenta is the only source in nature that you have two immunological systems working together, the mother and the baby, without any negative amplification, unless you suffer from preeclampsia or other pregnancy-related disorder. So the placenta was a good choice for a source of cell. But not only that, when we started back in 2005-6 to develop the product, on that time, the common understanding was that the mechanism of action should be differentiation and replacement, probably in the autologous manner, but if you think allogeneic, it doesn't look like a good approach to take cells, differentiate them, transplant them, and then you have to use immunosuppressive drug in order to avoid the rejection of the transplanted cells or organ. And then the idea was that we have to look for a different modality, to look for something that will be unique in the immunological properties on one hand, but on the other hand, will secrete a cocktail of protein based on need. And if you look into the placenta is a barrier between the mother and the baby, and actually give enough cells or ingredients to grow a baby from a fertilized egg to a full healthy baby within 9 months. So the placenta become the best choice for us. And for one placenta, we can produce 20,000 treatments, which contain each one of the treatments 300 million cells, which is a large amount of cells. So we started with the raw material, and then we looked for different products. So we took the same placenta, and we developed 2 products. Both of them are in clinic. The PLX-PED is from the maternal side of the placenta, the decidua, if you want, and the PLX-R18 is from the fetal side of the placenta. Looking into the mechanism of action, it looks very similar, but if you look into those mechanism of action of the PLX-PED on the left and for the PLX-R18 on the right, bottom right, you see different secretion profile. Even though both cells are from the placenta, they secrete a different cocktail of proteins and targeting a variety of different indications. So the PLX-PED for angiogenesis, muscle regeneration, and so, and the PLX-R18 for regeneration of dysfunctioning bone marrow, and the secretion profile that is presented there give you a feeling about what's going on. So looking into the process, after we make a decision that we want the placenta as a source, we know the conventional way of making cells. Each one of you understand that the way to do it, unless you can look for a different solution. And our solution was to grow them on 3-dimensional. Scaffold technology have been invented by the Weizmann Institute of Israel and the Technion of Israel, and we, Pluristem, acquired the technology from them back in 2007 and started to develop the process by understanding 2 things. The placenta source is from nature. So it's not our cells. The only way to convert them to become Pluristem cells is to understand that while we culture those cells in 3D macrocarrier environment, we actually change the cell's properties by inducing them to different environment. So you can see here, probably that is the one. Okay, you will have to believe me. So you can see on the right a list of genes that are associated. Okay, you can see here genes that are associated with a difference in expression, whether they are grown on 3D or 2D. And what we identified here is actually unique composition of matter that represent our cells. And not only that, by using the 3D macro carriers, we actually managed to be more efficient. So what you see here is that one bioreactor of 15 liter is equivalent to around 150 multi-trays that we have seen before. What is the meaning behind that? We can control better the process and we can become more efficient. So you can see here is a highlight of the manufacturing processes. So we start on the left with isolating the 2D cells from the placenta, and then we move them into our bioreactor system, which enable us to do the manufacturing as needed. The same manufacturing processes produce two products, the PLX PAD and the PLX R18. The only change we do is changing some ingredient during manufacturing and changing the control of the system. And then I would like to share with you some information, some experience we had during the process development. So what you see here is that we start to develop a process, and we call it low-tech and high-tech process. And as you probably hear from people that they are moving forward with the clinical studies, they are thinking about changing some culture conditions, some manufacturing method, and so on. In the beginning, it's looked very promising, because what you see here, that looking into the cytokines expression 1 or 2, the yield, the potency assay, everything looked promising. But if you dig a little bit further into the potency assays and post-hoc viability and other, you see a significant difference between those two processes, even though it looks to you exactly the same. So what to do? You have to think how to solve the discrepancies that are associated with the change in manufacturing processes. And then you have to think about changing the process, changing the ingredients to bring it to the level that you want, going back to the initiation of the development process, change the parameters that are required in order to make it happen. And you can see here a variety of parameters that have been tested along the line in order to see that any tiny change that you do in the manufacturing processes change the properties of the cells, change the secretion profile, change the potency assays. And why it's so important to mention, because in the early beginning, when we started the process, we put the slogan in place that in cell therapy, the process is the product, or the product is the process, depending how you want to phrase it. But the understanding that once you claim that cells are communicative organs and they can change the secretion profile as needed, you can extrapolate from that definitely they will change the properties if they will face any change in the culturing condition and so on. And we took a challenge upon ourselves. As you know, medium, most of the industry is using serum ingredients in order, serum from animals, in order to culture the cells and to provide them with the appropriate ingredients that are needed. And we took a mission upon ourselves to develop a serum-free product to ensure that we can bring the product to marketing. So you can see here some processes. You can see a variety of ingredients that have been in composition 1, 2, etc. in our product. And then we tested the serum-free product compared to the control, which was the serum product, looking into the right ingredients in order to ensure that in the end of the day, we can guarantee that the quality of the product will be identical, even though we changed the serum to serum-free. And in the end of the day, what we do, we test the product not only what are the culture parameters which are presented on the right upper side on the right of the slide, and then on the bottom of the slide, you can see the potency assays that we use, but not in vitro, but in animals. So whenever we do a new development, we go back to the animal models and we demonstrate that the changes we have done represent good potency in animals and not only in potency assay that is easier to achieve. So you have some hints to the one that are interested in upgrading manufacturing. And the purpose behind my presentation, or our presentation, is to give you some hint that when you think about manufacturing, better to start from the early beginning and then to do it step by step. It's a long process, but that the only way that in the end of the process, you can have a product that can be marketable and help patients. Thank you very much.
Thank you very much. So you just really in time, so unless there is a very burning question, I'm going to move to the next talk. Is anybody that really wishes to make a question? Okay, so we have there, please make it short.
Well, I think it's less a comment on it. I think it's very important to produce good products, but it's also very important to make quality control. And I think it is very important, especially for those, that quality control is not made by antibody-based technologies. So we have developed a Raman technology which allows to immediately see how cells react in their native status to any kind of drugs or goodies, as you mentioned. It would be very interesting to follow up what you saw in the moment using Raman technology.
Okay, you're completely right. You have to invest heavily into the QC, the quality control and quality assurance method. From each batch of our product, we contribute about 2 billion cells for QC. You cannot do it in autologous manner because of the number of cells that are required for a good quality control system. So that's what we pay today, 2 billion cells for each batch in order to guarantee that the quality is appreciated and achieved.
So I think it's time for a new technology which needs only a few cells and keep them as natural as possible without enhancing or changing their features due to producing a lot of cells for QC.
Theoretically, you are right. You can take one cell. In order to reconstruct a hematopoietic system, you have to have only one good progenitor cell. So technically, probably it can be done. The question is, can you achieve it in real when you want to treat a patient, to give him one potent cell, and to believe and to hope that the potent cells will proliferate and generate what is needed? I don't know the answer. Probably you have.
Thank you.
Okay, thank you very much again for the great talk. For the sake of time. So it's my pleasure to introduce Andrew Kaiser from Miltenyi Biotech. He's part of the managing team of the R&D at Miltenyi. His talk is entitled Pioneering Cell Processing of Personalized ATMPs.
Thank you, Sebastian. Good afternoon, everyone. Thank you also to the organizers. It's a pleasure to be here. And the aim here is going to be to continue to discuss automation and present you a little bit the capacities, the capabilities that we're trying to bring to you. So next. Yes. Thank you. Next. Yep. There you go. So what we do here is that we, as a tool and technology provider, we develop different aspects of the needed for the manufacturing of ATMPs, and we have devices, consumables, reagents, a lot of expertise, and we try to bring all of these together to have solutions that are as optimal as possible. The platform that we use is this Prodigy device, CleanMax Prodigy, and it has, let's say, all the features to do automated manufacturing of many different cell types. And I'm going to try to walk you through this today. And we often use the term closed, as it's a single-use tubing set that contains the cells, but you'll see that it's not closed in terms of how much you can actually manipulate the process to manufacture different cell types. The advantage of such platform, obviously, we just discussed about the changes along the way. You start to have to test a lot of different variables, but during your process to translate towards the clinic, you have to you have less and less chances to bring in changes. And the good thing with this platform is you can start to do the development in one platform, move it, and keep always the same. And this means you don't have these surprises to go from your manual, let's say, to your further to your automated process later on. So what we've done is we've worked very hard on a multitude of processes and rolled those out over the years. And you see these are some of the processes that we're working on. Of course, generate CAR T cells. I'll speak more about in a minute. We can also make virus-specific T cells, Treg enrichment. We're working on TILs, dendritic cells, stem cells, etc. And so this shows you that that one platform can actually be used for many different purposes. As an example, focus a little bit more on the CAR T cell here, and this is from a publication of a collaborator already several years ago, where they explained, their process with all of these different process units and devices that they have to use to manufacture their CAR T cells, and they published. We didn't ask them to, but we're pleased with this figure here, the prodigy that is covering all these different process steps. And this is another publication where we try to disclose a little bit more the details of the manufacturing also. So you saw at the beginning I was discussing this reagents, the device, our expertise, and you see here, for instance, where we're using all of the Miltenyi products here for the buffers, the medium, the activation reagent medium. We do CD4, CD8 enrichment that allows to normalize the starting material. We work mainly with lentiviral vector and developed this TS520 here for the manufacturing of CAR T cells. So you have all of these different steps in the process. We published that, so I'm not going to discuss the results here. And we just say there is no product without QC and in-process control. And so we also work very hard on having the workflow along the manufacturing to analyze the cells. And for this, we're also working on detection reagents, panels. We have our MaxQuant device allows for automated analysis or acquisition and analysis of the data. It can also be used for release. And just as an example, here is this staining reagent for CAR, for instance, CAR20. Here are these panels with automatic analysis. And we also have solutions to measure functionality of the cells. And as an example, just a short one, on a typical CAR T manufacturing process, with this enrichment, transduction, so activation, transduction, expansion, and formulation. This is part of the publication before. We showed all the way back then a relatively robust expansion of CAR T cells. And in the meantime, we transfer that to cell factories, clinical cell factory, for instance, at Miltenyi. And you see this is the type of reproducible manufacturing we get. And because it's on a prodigy, on a device with a program, you can actually very easily transfer this to other teams. And this is what we did here to transfer to other CMOs, and just as an example, some of the runs that they got. And, this went out much, much further, and publications are starting to do the work to show what's going on. So this is, for instance, in Spain, the use of the Prodigy device here to manufacture the CD19 CAR T cells at a point of care. Same thing, another paper here in US, and you can see this is 8 to 10 day processes here with IL-7/15. It's a frozen out product, but you can also use it as a fresh in, fresh out. This is in Moscow, 10-day process, and this is entirely serum-free, for instance. What we do is to try and support also, because as we did along the way, we developed this process. We did a lot of documentation and learned a lot, and we try to make this also available to all our customers. So we can support in terms of the manufacturing packages, the infrastructure package, even the device qualification, the QC flows and safety. And then, of course, we have a list of all the reagents. We can do trainings and support on regulatory support files. So I talked about CAR T cells, but I told you also the device allows for much more. And these are just another way of representing the types of cell types that we can expand in the Prodigy. And just to give you an example, here is also the HSCE, so the hematopoietic stem cell engineering process that we're working on and releasing, where we start on day 0 with CD34 cell enrichment, day 1 transduction, have a final product between day 2 and day 4, and you see with a one-shot MY30 here, an average transduction efficiency of 55%. But this goes on. We also allow the device to actually prepare adherent cell cultures. And now what this can do is the program allows to have a very flexible culture setup and interaction with external modules. In this case, for instance, a cell stack here, and you can see how you can scale towards adherent cell cultures. And the processes are quite well developed for mesenchymal stromal cells here, for pluripotent stem cells, and also for dopaminergic, progenitor differentiation here. So you've seen we keep on adding features to the device, and you've seen, for instance, this morning the catapult is using the electroporator device. And I would like you not to forget that we also have the Taito, which is a single cell sorter, which allows also to get very specific subsets. And you can also get that to work with a Prodigy. And on this, my last slide, I hope that I showed you through all of these technological advances how we're aiming to help patients through advancing scientific research and medicine by providing solutions for curative cell and gene therapy, as well as biomedical research. So on that, I'd like to thank you for your attention, and I'll take questions if there are any.
So again, we're perfectly in time, so we have time for a couple of questions.
There's one over there.
I have a question to your newest combination. How I know that in the Prodigy you do temperature and control in that little device. How do you do that with the stack? Is that stack then fused and defused and put into an incubator, or how is that done technically?
It is put in an incubator, indeed. Yeah.
You mentioned that your device is also supporting the pharmaceutical release procedure. Could you comment on that a bit more in detail?
Yeah, sure. So actually, the device itself, the Prodigy doesn't, but you can take samples, take them to the quant, and there, this automated analysis that we have, we call it express modes, will give you, based on certain panels and these qualified let's say analysis approach, will give you the transduction efficiency, viability, counts, composition that are used for release.
Do you have also solutions for microbiology testing?
No, so that's a good question, thank you. So actually, this is separate, right? So this is typically going to be on other devices, but we're working on ways to integrate all the information, for instance, on a single batch record. But no, this would be then, use of other devices. But there we can also help in that direction. Mm-hmm.
Hi, thanks for the talk. So I would like to ask about the culture vessel that you had in the device. Does it support suspension culture and microcarriers, etc., or is it just, let's say, for the T cell case?
So the T cells, obviously, we do suspension. Yeah. That's an absolute given. For microcarriers, it is possible. But actually, one of the experts who's tested this in-house is Sebastian over there. So maybe he can comment further on that. You want to, Sebastian?
So in theory, the system can support adherence cells, suspension cells. It's a matter of development of the different applications, I'd have to say. So it's in theory possible. So there's no extensive experience with microcarrier in the system as it is right now. But actually the system could support any kind of periphery that is driving such a culture, and is actually agnostic what you put on for the processes when you have an external device that drives it.
Yeah, because it could be more complex in terms of how you harvest your cells, etc., etc. So that's why I'm asking how you would deal with.
So the liquid handling could be supported by the device, and then you can outsource to an external cultivation vessel.
Okay.
And it really depends on the microcarrier. Some are digestible, some are not, right?
Yeah.
Some depend on temperature. So you,
you foresee to plug it in another bioreactor, and this would serve then as a downstream process unit, let's say then.
But integrated,
right?
yeah.
Okay. Thanks.
Thank you.
Okay, thank you very much once again. Thank you. So next speaker is Annelise Benesser from INSERM in France. She's going to speak on generation of hematopoietic cells with lymphoid potential from iPSCs for adoptive immunotherapy of cancer and leukemia.
Well, thank you very much for your presentation. So, no, doesn't. Green? Yes, it's green, but doesn't work anymore. It's okay. Okay, thanks. So, as you know, adoptive T-cell immunotherapy is currently so using autologous T-cell. What is going on? Okay. So, using autologous engineering mature T-cells are currently a very promising strategy. And as you know, there is we learned from the clinical trials that there is some key factors for durable clinical remission, and it is consistent to control the T-cell expansion and not to be exhausted, but also to have an optimal T-cell persistence of the CAR-T, for example, after the injection. And we now know probably that the less maturity of cells would be also more efficient. So with this, we asked the question if we could use T cells derived from iPS cells as novel source for new therapeutic approaches. And for the ultimate goals would be to generate a source of T cells and also could select different subset of cells from naive stage, stem cell memory or effective T cells from universal sources, but also to have and to provide off-the-shelf tumor-specific stabilized killer cells and also T cells tailored for any kind of application in medicine. So for this, our team has been working for 20 years ago now, first from murine embryonic stem cell and human embryonic stem cell, and now on iPS cells on hematopoiesis. For sure, we are now completely agree that we have a transient hemangioblasts, which is the first wave of the primitive hematopoiesis. We can now also produce immune cells, but also granular erythroid and megakaryocyte cells. But for instance, the main big challenge is currently is to identify primitive and let's say definitive hematopoietic stem cells with a self-renewal capacity and with a long engraftment, multi-lineage engraftment in vivo. And it has not been done currently. We cannot generate spontaneously this type of cells, except when we enforced some transcription factor, but also it requires probably permissive microenvironment, and so the vasculature is very important, and so promising technology will come from the 3D organoid system. And for example, as you can see here, we are able to produce completely in a dish, also in vivo, a structure of bone marrow from these iPS cells. The second challenge is also to identify very early progenitor able to common lymphoid progenitor able to entrance into the thymus and also to give rise of T cells. for our talk. And the second and the last one, can we mimic in vitro the thymopoiesis in order to select, to have inducing negative and positive selection of these thymocytes? So first, importantly, is to start with the right iPS cells. We have now currently more than 200 iPS in the lab, and we have screened different iPS cells, and whatever the reprogramming process we used, what we can see is that we have a high variability of these iPS cells regarding the capacity to produce progenitor hematopoietic cells, granular or erythroid cells, and as you can see, some of them are completely deficient in terms of this capacity, and also in terms of quality, some of them are very committed to the granular, or for some of them are giving rise erythroid cells. So it's very, very, very variable. And so we ask the question, what is the reason first? And if you can have a markers, very early markers at the pluripotent stage, if we can predict this behavior of this IPS during the culture. So we use global microRNA expression profiling by analyzing more than 100 microRNA, and we confirm the existence of one microRNA 206, which was overexpressed in cells which were deficient for hematopoietic deficiency. And when you overexpress this microRNA in a competent embryonic cells H1, you can see we have a higher decrease of the colony assay in the colony, and also the percentage of CD34 positive cells. And the important thing is there is two targets of this microRNA. It is RANKS1 and TAL1, which has both are very master transcription factors that working very early at the hemangioblast stage. So now we are selecting these cells, and so we are going to have a robust T cell differentiation protocol. First, we have to induce a mesoderm by the embryonic bodies technology. This is aggregate with different factors, and BMP4 is a very important factor from the beginning. Secondly, we have to have a specification of this mesoderm into the hematopoietic lineage, and during this process, we have with MS5 and different cytokine, we track different progenitors with a lymphoid commitment, and we identify very immature hematopoietic progenitor cells with a marker of CD45RA positives on the membrane. And as you can see, they express different transcription factor, Icaros, small amounts of RAG. Also, we have also pre-T alpha. But as you can see, it is also a mix with different myeloid cells. So we have also to decipher within this population, different subpopulation, and we are going on with a single-cell RNA sequencing. So last step would be to mature these progenitor cells using two strategies, using stromal cells expressing notch ligands, delta-1 or delta-4 with cytokines, or using the thymic or microenvironment with a 3D thymic culture system to induce a positive selection. So we asked first the question, which if this progenitor, immature progenitor, could entrance into the thymus. So we used a thymic colonization assay with the CFS surface labeling, and we can confirm that the subpopulation of cells expressing CD7 could entrance into the thymic using thymic lobe from embryonic embryos from mice, and these cells are expressing some of that expressing CXCR4. So a recent paper have confirmed that during the long-term culture system on a 3D stock using this technology, we can identify a post-thymic immigrant cells, which is very closely to naive T cells, and we have single positive CD34, CD4 and CD8 alpha beta, TCR alpha beta. and they're performing a repertoire, a very diverse repertoire using on these in this culture system. So, in the extractimic progenitor, we can have also we can use also the stromal cell with notch ligand. With a 2D culture system, we can confirm that we don't have single positive CD8 cells in vitro. We are just in an immature fetal T cell innate-like cells with a double positive thymocyte with RAG expression. And if you want to have knife-like cells, you used a 3D organoids aggregate stromal cells, and it's the last paper calling now as an artificial thymic organoids. So, for instance, it is a unique paper claiming that we can use this technology to mimic the thymus. So we have to optimize the differentiation in feeder-free condition and to also improve maturation. And now there is some paper to bioengineering thymus organoids. So in terms of strategy, based on this technology, we have two possibilities: to reprogram the T cells into iPS, introduce a CAR T, and then again produce T cells with a CAR. And so the seminar work from Sadlin confirms that these very immature T cells can eliminate Burkitt lymphoma in the not-skid mice, and there is a very impact on the survival. Second possibility is to regenerate exhausted T cells by reprogramming technology. So you take some exhausted T cells, you reprogram these cells, so you have the rearrangement endogenous TCR inside, and so you reproduce T cells. They will lose the PD-1 expression, but during this time, as you know, you have a recombination. So you have to also eliminate also the endogenic rearrangement, so you can also have a RAG2 KO to prevent GVHDs and to stabilize antigen-specific TCR during this process. So for finish, we are now set up an iPS cell therapy platform for the program Haplobank, meaning that we have to build now the starting material of iPS HLA matched and knockout for HLA. And for this, we used the Prodigy Clinicax Prodigy for the first beta test. So we test the Prodigy to amplify these cells in a CCU, 1 cell stack and 5 cell stack. And during this process, we can see we have a huge expansion with a billion of iPS cells after 70 days. And we have a very stability of the pluripotency markers. We have a popping doublet is completely the same in the process or in vitro with a normal karyotype. And there is also a pluripotent with StemMax kit differentiation. So for finish, I want to thank the team, the postdoc and students which has participated on this work, and also CONIF, which has we are collaborating in Canada. Thanks.
Thank you very much, Annelise. So we have time for maybe one question. Anybody? Okay. If not, we are just in time. Thank you once again. Thank you very much.
Okay, so I'd like to announce our last speaker of the session. It's Nuno Neves. He's principal investigator at the University of Minho in Portugal. And his talk is entitled Functional Surfaces and Scaffolds Combined with Stem Cells for Bone and Cartilage Tissue Engineering. Please welcome.
Thank you so much, Mr. Chairman, for the kind presentation. I would like to start by acknowledging the organizers for having me talking today. And I will start by showing the motivation of the work that we are doing in the lab. So basically, as you know, when you lose cartilage, you lose it for good. So basically, the end stage solution that we have for this kind of process is basically to use implants that by 90 to 95% of the cases are very successful, but by 10 years, they are not very effective. So they need to be revised. So, there are already in the market some solutions for this using advanced therapies, but as you know, there are issues of inconsistency in the results and also of reimbursement. So the two works that I'm showing to you today kind of contribute for this problem of cartilage regeneration. So basically, the first work that was done by Marta Casanova in the lab, which is a PhD student that is kind of finalizing, is the chondrogenesis in the inductive nanofiber substrate using both biological fluids and mesenchymal stem cells from an autologous source. So the idea here is basically to use antibodies at the surface of these substrates, which are nanofibrous electrospin polycarblactone substrates that we modify at the surface with antibodies that are able to capture growth factors from platelet lysates. And the idea is really to demonstrate that that strategy can really drive the mesenchymal stem cells into the chondrogenic phenotype. So the idea here is we modify the surface with UV ozone, and then we promote amine groups at the surface, and then we use those amine groups to immobilize the antibodies at the surface, and we block the amine groups that are not used. And then we dip this substrate into either a solution of recombinant protein or the platelet lysates, and then we look for the selective binding of growth factors of interest. So in this case, we use IGF and TGF-beta3, and basically, we see human bone marrow stem cells at this surface, and then we look for the chondrogenic differentiation. As positive control, we use the chondrogenic induction medium that is very well known. Now, when you look for the results, if I have them, maybe you can press. Okay, that's very good. So thank you so much. Here we have the fluorophore that is able to show that we have anti-TGF-beta3 and anti-IGF-1 at the surface, and they even co-localize into this kind of yellow. This is the non-specific kind of interaction. Then we look for the 3 different donors for the amount of TGF-beta3 and IGF-1, and as you can see, there are different amounts of these growth factors present in platelet lysates of different donors, and we typically pull those platelet lysates in order to have a more controlled kind of source of the growth factors. And we have a very large immobilization efficiency, in this case 99.3, and in this case 77.5. Now, when we look for the biological activity of the growth factors at the surface, indeed we see that we don't have any issues of cytotoxicity. The cells produce protein, and they have mitogenic activity, and they produce GAGs in increased amounts. And it seems that we have a slightly, more effective biological activity of the growth factors coming from platelet lysates as we compare them with recombinant. We looked as well for the chondrogenic differentiation by Alshan blue staining, and this is our control. And as you can see, the different conditions, either from recombinant growth factor or from platelet lysates, both of IGF and TGF-beta3, or the combination of both, they are effective. And it seems that even IGF-1 in itself is very effective in driving the chondrogenic differentiation. Now, we looked as well for chondrogenic genes like SOX9, collagen type 2, and aggrecan, and as well for this negative gene. And we see that we have very low values of collagen type 1. We have a consistent expression of SOX9, collagen type 2, and aggrecan, which is very positive. And we also use immunocolocalization to demonstrate that we have indeed the production of collagen type 2 after 28 days in increased levels, especially for the IGF-1 condition. So just to summarize, what's the idea here? The idea is to use this substrate in combination with a microfracture surgery. And the idea is really to use these growth factors at the surface to drive the chondrogenic differentiation of stem cells. Now, another way of using this strategy of immobilizing antibodies is to use these nanoparticles in such a way that we can immobilize at the surface antibodies that are able to capture pro-inflammatory cytokines in order to preserve the matrix of cartilage. And this work was done by Ana Claudia Lima in the lab. And the concept here is basically to inject locally in the synovial fluid nanoparticles that are able to capture interleukin-6, which is very central in this inflammatory process that develops in osteoarthritis. And the idea is to break this cycle of production of pro-inflammatory cytokines that lead to production of enzymes that degrade the cartilage. So this is the concept. Now let's look at a bit more details about the nanoparticles. So we use chitosan and hyaluronic acid with a high molecular weight, which is anti-inflammatory. And the idea is to use polyelectrolyte complexation to produce the nanoparticles and then to stabilize them by EDC and HS chemistry. So this is the way they look like. So they are very, uniform in size, and the polydispersity index of those nanoparticles is very good. And the zeta potential is not neutral, so it's very good to avoid agglomeration over time. And this is by EFM, the way they look like. We looked as well for the stability over time, and we see that after 6 months, they basically maintain the same size, which is a very good perspective. Now, we used as well EDC and HS chemistry to immobilize the antibodies at the surface of the nanoparticles. And then basically we quantify the amount of interleukin-6 that we are able to capture, and we demonstrate that we have really high values of interleukin-6 that we can capture by this strategy. But the point is, are we really able to inactivate the pro-inflammatory cytokines by this strategy? And I would like to pass the slide. Can somebody help me? Okay. Now, this is human articular chondrocytes when they are exposed to nanoparticles in different concentrations. And you can see that the cell viability is not affected by the amount of nanoparticles that we expose them to. And when we look for cell proliferation, indeed, after, above 50 micrograms per ml, indeed, there is some negative effect to the chondrocytes by the presence of the nanoparticles. Now, we looked as well for what the chondrocytes look like in 2D culture. And you can see that when they are exposed to increased levels of the nanoparticles, they keep a monolayer at the surface, which is a very good indication that we don't have cytotoxicity. Oh my God. This is not nice. Okay. Now, one very important point is that we don't want the chondrocytes to internalize the nanoparticles. So what you are seeing here is the chondrocytes themselves, and then with 2 different amounts of the nanoparticles with a fluorophore. So you can really see that they stay out of the chondrocytes, and this is a very important point because we want them to be in the synovial fluid capturing interleukin-6. One very important point is to use as well monocytes, in this case THP1, which is a cell line of monocytes. And when we expose them to different amounts of the nanoparticles, they keep having the same properties, so they don't show cytotoxicity. And when we use primary monocytes, we don't see as well any kind of cytotoxicity from the nanoparticles. Now, we want as well to show the functionality in capturing interleukin-6. And what you are seeing here is when we expose human articular chondrocytes to a conditioned medium of activated macrophages in a kind of a model of inflammation in vitro. So what we see is that when they are not exposed to the conditioned medium, they don't produce interleukin-6, but the chondrocytes themselves, when they are exposed to this conditioned medium, they start producing interleukin-6. When we use the free antibody, we kind of recover a little bit and lower the amount of interleukin-6 produced by the chondrocytes, but not in a stable way as we can get with the nanoparticles. So this kind of shows the functionality, and most probably we need to increase the dose that we are using in order to lower down this interleukin-6 level. So I hope I convinced you that the surface functionalized scaffolds support primary adult stem cell viability, expansion, and chondrocyte differentiation in this case. And the antibody mobilization strategy can be used in different ways in order to have biological functionality from the system. I would like to acknowledge the team, the funding, and of course Restore for having us here. And I would like to announce this as our winter school if you are interested. So with that, thank you so much.
Thank you very much for the talk. Are there any questions from the audience? Could you wait a second, please?
What is a way of binding to nanoparticles of the antibody?
You mean the dose of the antibodies that
No,
we
how
are
you
using?
actually attach antibodies to
How
the
do I
nanoparticles?
attach? So
Yes.
by EDC and HS chemistry, this is a very common way of using a kind of a linker that we can use to the surface in order to activate to use the antibodies at the surface.
All right, but
and we
my
can control the dose that we can use on there. Yeah.
All right, my question is it site specific or your antibody could be orientated in a wrong way? For example, FC fragment.
That's a very good question. Thank you so much. Yes, indeed, we can use antibodies that are able to capture and keep the bioactivity, as I shown in the first work, but we can tailor the antibody in such a way that is able to capture and inactivate the bioactive molecule that's of interest. So we can really fine-tune the antibody in order to have a specific bioactivity at the surface. In this particular case, the idea was to capture interleukin-6, but we have a follow-up work in which we used antibodies into different pro-inflammatory cytokines, and we are showing the usefulness of this strategy, not only in vitro, but as well in vivo. Okay.
I have a question regarding the uptake or the potential uptake of your particles. If you think about an in vivo situation, you would add a particle, it would maybe induce phagocytosis or other processes, and maybe you can comment on this?
For sure. In fact, we have just completed an in vivo study in which we injected locally in the synovial fluid the nanoparticles. What we see is that indeed we have a functional protection of the cartilage. All the indications that we have in the in vivo study show that indeed the nanoparticles are able to protect cartilage, and we don't see any toxic effect or any functional effect coming from the nanoparticles in the synovial fluid. That said, of course, we need to make sure that we don't have the nanoparticles going into the bloodstream and eventually staying in other organs. So this is a follow-up in vivo work that we need really to do to show that we don't have any toxicity out of the synovial fluid. Thank you so much.
So I may have another question concerning over here, to your left.
Oh, okay.
And to the first part of your talk and the functionalization of the fibers. You showed that the chondrocytes adopt a collagen II phenotype, and after you use that in vivo, would that be maintained, or is there a tendency of ossification later?
If I understand correctly, the issue is about collagen type 1. Versus collagen type 2.
The collagen type 2, yes. So will it turn into one?
In fact, this is one of the difficulties when we use stem cells for cartilage repair, because it's very difficult to ensure that the phenotype of the chondrogenic differentiation is stable. In fact, those cells, as we know, tend to go into the osteogenic phenotype by hypertrophy. So the idea here is really to have a system that is able to promote the differentiation of stem cells, especially in this context of the microfracture, or else some other technique in which we can supplement, the defect that we have in cartilage with some cells. And the idea is really to use this substrate to drive the cells into the right phenotype, not really fibrocartilage, but a stable cartilage phenotype that is able to restore the cartilage defect. So this is really the concept. Of course, we need to demonstrate this in vivo. Yeah.
Okay, so if there are no further questions from the audience, I'd like to thank all speakers of the session and like to end the session. And the next sessions are now the breakout sessions. Thank you all for attending.
So just an information to you, though, The breakout session here in the plenary room will be about preclinical models and technologies, with a focus on the human on the chip. If you are interested more in foundational research, new targets, new technologies, this will be in the room Berlin, which is just opposite this plenary room. And in case you are interested in manufacturing technologies, including product characterization and automation, this is in the London room, just behind the Berlin room. And for sure, for everybody, we will have later on or in between of the breakout sessions, we will have a coffee break, which will be at 10 minutes past 5.
But also the point was the point